Hello everyone, my name is Weihan and I'm a fourth-year PhD candidate in the US under Brian Kennedy's lab. So today I'm going to show you my research as well as my understanding of aging. There's a simple goal for my research, which is to extend the lifespan of human beings further, and further. But the question is that how do we extend the lifespan of human beings? Now, the first thing we need to ask is, what is aging? So I want to use this spring with a ball attached as an analog. So now this is a simple spring and we call it a simple system. Similarly, our body is another kind of system. So now this system is in its equilibrium state because it's not changing. If you pull this ball a bit, it will bounce back to equilibrium. So this means that this system is stable and resistant to be unstable. Similarly to our body, we have the same way. So tomorrow we wake up, we look the same as yesterday. And then also, if we have a disease, we will get recovered. A disease can be considered as like you're pulling the balls. And then once you remove the force, it bounces back. But just remember that this is a stable system in a short period of time. But as time goes by, what will happen? So there are many factors that actually cause this spring to lose its resilience. For example, metal fatigue, right? Similarly, in our body, we also have these things that we cannot bounce back as time goes by. So if we're considering these things that can be bounced back as homeostatic events, because in biology we call it homeostasis, it is in equilibrium. So there are factors that are non-homeostatic. For example, stem cell exhaustion, telomere shortening, and protein aggregation at a certain extent, because we know that Alzheimer's disease has these protein aggregations. So now the question is that if we know that there are these factors that can drive aging or the hallmark of aging, how can we actually extend the lifespan of human beings? Remember that there are non-homeostatic events which are driven by the homeostatic events. Can we actually remove these non-homeostatic events? But the thing is that in biology, it's not something as simple as a spring. There are so many hallmarks of aging, and they're actually driven by some really simple biological pathways. But this represents actually a systematic change in your body. So if we think about it, your body as a system, and there is a systematic change, so we are looking for a systematic intervention. So how do we do this? My idea is that we consider our body as a homeostatic network, and then with these genes clustered as modules within, and then we will ask the question, at what condition is this network or system long-lived? So they can be under different perturbations like genetic interventions or drug interventions. So if we can collect these data in, and then we will get a answer of, and then we will basically ask, what does these modules behave in long-lived systems? And then we can get these modules that are associated with longevity, so that any interventions that can extend the lifespan are expected to have their impact on these modules, so that we can use them, go back to look for the drugs or other kind of interventions that can extend the lifespan of human beings or any model species. So here is a compound that I predicted from my modules, and now we can see that at 20 micromolars, it can significantly extend the lifespan of the median and maximum lifespan of C. elegans. Beyond the discovery of novel neuroprotectors, I'm also working on predicting drug pairs, combinational interventions that can further extend lifespan of model species and potentially human beings. And this is an ongoing project, which I'm not going to cover a lot about it, but currently we have a lot of success compounds that are already being predicted based on my algorithms. Even though I cannot disclose their names at this moment, so one of them is rapamycin, but you can clearly see that when you combine these two drugs, you're getting a further lifespan extension compared to the individual one. And this is actually achieved by some transcriptomic level prediction. After we predicted our compounds, I'm going to test them in the model species. The most commonly model species we use is called C. elegans, which is a small tiny worm, but with a relatively short lifespan. The cool thing about the C. elegans is that even though it's a tiny worms and genetically distant from human beings, there are a lot of biological pathways that are preserved between C. elegans and human beings. And then especially for the aging pathways, such as mTOR signaling pathways. So in my spare time, I like to play video games, and also I like to play Weiqi, an ancient Chinese chess. And I'm very good at it, and here you can see are my trophies. Thank you for your listening. And if you want to learn more about my research, or you want to have any collaboration with me, you're welcome to contact me through my email address. Thank you.